We often credit our greatest achievements to planning and intellect, but history is also shaped by serendipity, the happy accident. In medicine, some of the most profound breakthroughs have come not from careful design, but from unexpected mistakes. A forgotten experiment, a simple error, or a moment of carelessness has sometimes led to discoveries that changed the world. These are stories of the prepared mind meeting the unexpected and having the wisdom to see its potential. Join me as we explore how accidental moments have transformed healthcare and saved countless lives. These are medicine's accidental miracles, proof that sometimes the greatest leaps forward are stumbled upon. Our first story begins in 1958, a time of burgeoning scientific discovery at the University of Buffalo. It was there, amidst the hum of equipment and the scent of solder, that engineer Wilson Greatbatch was diligently working. His mission? To build a device, cutting edge for its time, designed to record the intricate rhythms of the human heart. Deeply focused on his intricate work, surrounded by a myriad of components, he reached for a resistor. In a moment of unintentional selection, he accidentally grabbed a 10 megaohm resistor instead of the 1 megaohm resistor he intended to use. A simple mistake, one might think, but this seemingly insignificant error would alter the course of medical history. When he powered up the circuit, expecting the steady, predictable hum that should have filled the lab, he was met with something entirely different. He didn't hear a steady hum. Instead, he heard a rhythmic pulse, thump, 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 a sound mimicking the very essence of life. It was, in essence, an artificial heartbeat generated by accident. Greatbatch instantly realized the profound significance of what he was hearing. This device, born from a mistake, could potentially regulate a failing heart, offering a lifeline to those in desperate need. At the time, pacemakers were far from the sleek devices we know today. They were bulky, external machines, cumbersome and restrictive. These machines tethered patients to hospital beds, limiting their mobility and carrying significant risks associated with their invasive nature. Greatbatch envisioned something radically different. A small, implantable device, discreet and self-contained. A battery-powered guardian, small enough to reside within the body, that would restore a heart's rhythm only when needed, offering freedom and a new lease on life. He dedicated the next 2 years to refining his accidental invention, meticulously working to perfect its design and functionality. He understood the importance of collaboration and partnered with Dr. William Chardak to ensure the device was not only effective, but also safe for implantation in the human body. Finally, in 1960, the moment arrived. A 77-year-old man suffering from a complete heart block, a condition where the heart's natural pacemaker fails, received the very first implantable pacemaker, a groundbreaking moment in medical history. The result was immediate and undeniable. His heart beat steadily. With a newfound rhythm and strength, his life was irrevocably transformed. He lived another 18 months, a testament to the device's effectiveness and the renewed vitality it provided. He was freed from the constant threat of collapse, able to enjoy his remaining time with a sense of security and peace. This accidental invention, this fortunate mistake, launched the entire field of cardiac rhythm management, revolutionizing the way we treat heart conditions. Today's pacemakers are tiny, intelligent marvels of engineering, constantly evolving to meet the needs of patients. They are life-saving devices for millions around the globe, offering a chance at a longer, healthier, and more fulfilling life. All of this began with a simple mistake, a seemingly insignificant error in the lab. It was a misplaced resistor and a mind that was open and ready to see the potential hidden within the unexpected outcome. Greatbatch's error became a miracle, a testament to the power of serendipity and scientific discovery. It proves that sometimes the most profound innovations are sparked by the unexpected, by embracing the unforeseen path. Millions now live full and active lives, their hearts beating reliably and strong, thanks to this remarkable device. All thanks to that fateful day in the lab when a simple mistake led to a life-changing discovery. The legacy of that one error is immeasurable, rippling through generations and transforming countless lives. It's a testament to the power of human curiosity, the importance of observation, and the willingness to embrace the unforeseen detours on the path to innovation. The story of the pacemaker serves as a powerful reminder. Not all breakthroughs are meticulously planned or strategically executed. Some of the most impactful discoveries are stumbled upon by accident. And sometimes, that unexpected twist, that accidental finding, is exactly what the world desperately needs to solve its most pressing challenges. The accidental heartbeat became a lifeline for generations, a symbol of hope and resilience for those facing heart ailments. All because one engineer, driven by curiosity and a desire to improve lives, reached for the wrong part, forever changing the landscape of medicine. Our next story takes us back to the late 19th century, a time of burgeoning scientific inquiry and groundbreaking discoveries. Specifically, we find ourselves at the esteemed University of Strasbourg, a hub of intellectual activity and rigorous research. It was here, within the walls of this institution, that two scientists, Oskar Minkowski and Joseph von Mering, embarked on a series of experiments focused on understanding the function of the pancreas. Their initial goal was to study the role of the pancreas in digestion, and to that end, they performed a procedure to remove the pancreas from a dog. However, in the days that followed, they began to notice something quite unexpected, something that deviated sharply from their anticipated results. The dog's urine, rather unusually, started attracting swarms of flies, an observation that piqued their curiosity. Instead of simply dismissing this peculiar phenomenon as an irrelevant anomaly, Minkowski, driven by scientific curiosity, decided to investigate further. Upon closer examination and analysis, he discovered that the dog's urine was saturated with sugar, a clear indication that the animal had developed diabetes as a consequence of the pancreas removal. This seemingly accidental observation, born out of an experiment designed for a completely different purpose, established a crucial link between the pancreas and the onset of diabetes. This was a monumental discovery, shedding light on a disease that had long remained a mystery, baffling doctors and researchers for centuries. Their initial experiment, meticulously designed to unravel the complexities of the digestive process, unexpectedly revealed the pancreas's absolutely vital role in the regulation of blood sugar levels, a function previously unknown. Decades later, this groundbreaking discovery served as the spark of inspiration, igniting a new wave of research and investigation. It motivated Frederick Banting and Charles Best, working tirelessly in their Toronto laboratory, to embark on a quest to isolate the elusive sugar-regulating substance produced by the pancreas. After numerous trials, setbacks, and unwavering dedication, they finally achieved their goal, successfully extracting insulin from the pancreas. This extracted insulin proved to be a game-changer, dramatically reducing blood sugar levels in diabetic dogs, offering a glimmer of hope for human patients. In 1922, a young, critically ill boy named Leonard Thompson, suffering from severe diabetes, was chosen to be the first human to receive an injection of this newly discovered insulin. The results were nothing short of miraculous. His dangerously high blood sugar levels returned to a normal range, stabilizing his condition. As his blood sugar stabilized, he began to regain his lost strength and vitality, showing remarkable improvement. For the first time in history, diabetes was no longer an inevitable death sentence, offering hope and a chance at a normal life for countless individuals. This monumental breakthrough, which has since saved millions of lives around the globe, had its humble beginnings in a seemingly insignificant observation. A curious scientist noticing an unusual number of flies buzzing around a lab. It serves as a powerful reminder that sometimes the smallest, most overlooked details can hold the key to the biggest, most impactful discoveries. The path that led to the discovery of insulin was not a straightforward one, filled with challenges and unexpected turns, but it was ultimately paved by the principles of open-mindedness, relentless curiosity, and the courage to thoroughly investigate the unexpected. From a simple observation of a dog's urine to the development of a global cure, the story of insulin stands as a remarkable triumph of accidental observation and scientific perseverance. In the realm of science, every detail holds significance, especially those that initially appear to be inconsequential or unexpected. The sweet scent of discovery, in this case, irrevocably changed the fate of millions of people living with diabetes. It all began with a simple yet profound question. Why are there so many flies attracted to this particular sample? The answer to that question unlocked a new era in the field of medicine, revolutionizing the treatment of diabetes. Sometimes the key to unlocking a life-saving cure is hidden in the most unlikely and unexpected of places. And it all started with a swarm of flies, a testament to the power of observation and the importance of pursuing even the most peculiar of leads. Our final story takes us across the Atlantic to London in 1928, a bustling metropolis, a hub of innovation, and the backdrop for a discovery that would change the course of medicine forever. We find ourselves in the famously untidy lab of Dr. Alexander Fleming, a place where scientific curiosity thrived amidst apparent chaos. Fleming was returning from a well-deserved holiday, eager to get back to his research. Little did he know, a serendipitous event awaited him. Upon his return, Fleming noticed something peculiar. A petri dish, one of many he had been working with, was contaminated with mold. Most scientists encountering such a setback would have simply tossed it aside, dismissing it as a failed experiment. But Fleming, driven by an insatiable curiosity, decided to look closer, to examine the anomaly. And what he saw was remarkable. Around the mold, the bacteria he had been cultivating had been mysteriously destroyed. He observed a clear zone of inhibition, a testament to the mold's potent antibacterial properties. It was then that Fleming realized the mold was producing a substance, something extraordinary. This substance was lethal to bacteria, effectively killing them, but remarkably, it appeared to be harmless to human cells. Intrigued and excited by his findings, he meticulously documented his observations and named this revolutionary substance penicillin. Though his groundbreaking discovery was initially overlooked by the wider scientific community, its potential remained untapped, waiting for the right moment. The outbreak of World War II, with its devastating toll of wounded soldiers and rampant infections, created a desperate need for new and effective treatments. It was then that scientists Howard Florey and Ernst Chain, recognizing the significance of Fleming's earlier work, stepped in. They revisited Fleming's research, meticulously studying his findings and recognizing the immense potential of penicillin. Together, they embarked on a mission to develop methods for the mass production of penicillin, a feat of scientific ingenuity and perseverance. Finally, in 1942, they treated their first human patient with penicillin, a pivotal moment in medical history. The results were nothing short of dramatic, a testament to the life-saving power of this new drug. However, the initial supplies were limited, and they quickly ran out, highlighting the urgent need for increased production. But by D-Day, enough penicillin was finally available to treat all Allied soldiers wounded in the invasion, a crucial factor in saving countless lives. Penicillin revolutionized medical care, turning previously fatal infections into treatable conditions, offering hope where there was once despair. It played a vital role in saving thousands upon thousands of lives, both on the battlefield and in hospitals around the world. Fleming's accidental discovery ushered in the age of antibiotics, a new era in medicine that would forever change the way we combat bacterial infections. It transformed medicine and dramatically extending life expectancy worldwide, impacting global health in profound ways. Diseases like pneumonia, once a leading cause of death, and meningitis, a terrifying infection of the brain, became treatable, offering hope and recovery to countless patients. And surgery, once fraught with the risk of infection, became far safer, paving the way for more complex and life-saving procedures. All of this began with a single contaminated petri dish, an unexpected twist of fate that would alter the course of history, and a scientist curious enough to investigate, to look beyond the apparent failure and see the hidden potential. The world changed, all because Fleming didn't throw away a ruined experiment, because he recognized the value in the unexpected. The age of antibiotics was born from a fortunate contamination, a reminder that sometimes the greatest breakthroughs come from the most unexpected places. Sometimes the greatest revolutions start with a simple act of curiosity, a willingness to question, to explore, and to challenge conventional wisdom. Fleming's mold became humanity's shield against infection, a powerful weapon in the fight against disease. The story of penicillin is a testament to the power of noticing what others overlook, of finding value in what others discard. One petri dish, one moment of attention, and the world was never the same. All because Fleming paused and said, "That's funny," a simple phrase that sparked a revolution in medicine. The pacemaker, insulin, penicillin, three pillars of modern medicine, each born from serendipity. History is often shaped by chance, by unexpected turns and unforeseen circumstances, not always by grand design or meticulous planning. Sometimes the most significant advancements arise from the most unlikely of situations. In 1928, Alexander Fleming's lab at St. Mary's Hospital in London was a mess, a testament to a mind deeply engaged in its work, perhaps a little too engaged to maintain perfect order. Petri dishes everywhere, stacked haphazardly, a landscape of ongoing experiments and forgotten samples. Experiments left unattended, cultures growing beyond their intended observation period, a scene of scientific exploration bordering on chaos. Fleming, brilliant but untidy, a man more focused on the microscopic world than the organization of his own workspace. He left some Staphylococcus cultures out, exposed to the open air, while he took a well-deserved holiday. Little did he know, this simple act of negligence would change the course of medical history. Returning from his break, he noticed something peculiar, a petri dish with a distinctive blue-green mold flourishing within it. This mold was surrounded by a clear ring, a halo of emptiness in a world teeming with bacteria. No bacteria grew near it. The mold had somehow created a zone of inhibition, a microscopic battlefield where it reigned supreme. Most scientists encountering such contamination would have dismissed it, tossed the dish into the autoclave without a second thought. But Fleming's curiosity, his innate scientific inquisitiveness, stopped him. He saw potential where others saw only failure. He carefully examined the mold, eventually identifying it as penicillium, a common type of fungus. He then embarked on a series of experiments, meticulously documenting his findings, and ultimately named its bacteria-killing substance penicillin. Unlike other chemicals tested before, penicillin possessed a remarkable ability. It selectively killed harmful bacteria, but crucially, it left human cells unharmed, a characteristic that set it apart and made it a potential life-saving drug. This was more than just another antiseptic. This was the first true antibiotic, a revolutionary concept in medicine. It was a substance that could target and kill invading microorganisms without inflicting damage on the body's own defenses. The discovery was pure serendipity, a fortunate accident born from a confluence of unlikely events. It began with a mold spore drifting in on the breeze, an invisible traveler carrying the key to a medical revolution. Then there was a forgotten dish, a neglected experiment left to its own devices, a breeding ground for the unexpected. And finally, a scientist who paid attention, who recognized the significance of the anomaly, who dared to question the ordinary. Fleming's messy habits had by accident created the perfect conditions for this serendipitous event to unfold, and in doing so, changed the future of medicine, ushering in a new era of infection control and saving countless lives. The world's first antibiotic was born not from order, not from sterile precision, but from chaos, from the unpredictable dance of nature and circumstance. The miracle of penicillin began with a simple oversight, a moment of inattention that blossomed into a medical revolution. Sometimes the greatest breakthroughs, the most transformative discoveries, come from the most unexpected places, hidden in plain sight, waiting to be recognized. Fleming's open mind, his willingness to explore the unknown, made all the difference, transforming a potential failure into a monumental success. A forgotten petri dish, a symbol of scientific neglect, would soon save millions of lives, a testament to the power of chance and the importance of a curious mind. Fleming published his findings in 1929, but the world barely noticed. He saw penicillin as a possible antiseptic, not a systemic cure. The scientific community overlooked his modest paper. Penicillin was hard to produce and unstable, making it nearly impossible to use widely. Fleming and his small team struggled to purify it, but the chemistry was beyond their reach. Frustrated, Fleming moved on, and penicillin faded into obscurity. For nearly a decade, its potential was ignored. The miracle cure sat in a forgotten journal, waiting for someone to realize its value. Deadly infections continued to claim lives, while penicillin gathered dust. The world was unaware that a cure was within reach. All it needed was the right team to bring it back to life. For 10 years, penicillin was a scientific footnote. Fleming had made the discovery, but he wasn't the one to turn it into medicine. The world still desperately needed a weapon against deadly infections. In the late 1930s at Oxford, a new team took up the challenge. Howard Florey, a skilled organizer, and Ernst Chain, a brilliant biochemist, led the effort. They were joined by Norman Heatley, whose ingenuity would prove vital. Their interest in natural antibacterial agents led them back to Fleming's forgotten work. This team had the vision and expertise to do what Fleming could not. The baton had been passed and the race to save lives was on. In 1937, Ernst Chain rediscovered Fleming's paper and saw its potential. He convinced Florey to pursue penicillin, and the Oxford team began their quest. They struggled to grow the mold, using whatever containers they could find, even bedpans. After months of trial and error, they produced a crude, unstable penicillin sample. Norman Heatley developed a new extraction method, making the drug more stable. By 1940, they had enough purified penicillin to test on living creatures. The team had overcome immense chemical challenges with limited resources. Their perseverance turned a scientific curiosity into a real, usable substance. The Oxford lab buzzed with anticipation. The next step would determine if penicillin could truly save lives. The world's first antibiotic was ready for its first real test. The Oxford team, driven by an unwavering commitment to find a cure, knew they were on the cusp of something extraordinary. Their big test, the moment of truth, came in May 1940. It was a pivotal point in their research, a chance to prove that penicillin held the key to fighting deadly infections. To rigorously test their hypothesis, 8 mice were carefully selected and injected with a lethal dose of deadly bacteria, a strain known to cause rapid and fatal infections. In a controlled experiment, 4 received the experimental penicillin treatment, while the remaining 4 did not, serving as a control group to accurately measure the drug's effectiveness. The untreated mice, left to succumb to the infection, died quickly, a stark reminder of the bacteria's devastating power. But in a stunning turn of events, the penicillin-treated mice not only survived the infection but thrived, displaying remarkable resilience and vitality. The result was undeniable, a clear and decisive victory for penicillin. The experiment proved, beyond any doubt, that penicillin could effectively cure a lethal infection in a living animal, a monumental achievement in the fight against bacterial diseases. The team was ecstatic, their tireless efforts finally bearing fruit. But amidst the celebration, new challenges loomed, casting a shadow over their triumph. Treating a human, with their significantly larger body mass and complex physiology, would require a vastly greater quantity of penicillin. The amount needed was far more than treating a mouse, presenting a significant hurdle in translating their success to human patients. Recognizing the urgency, the team scrambled to scale up production, working tirelessly to find ways to increase their output. They transformed their lab into a makeshift factory, improvising and adapting existing equipment to meet the growing demand. Every available container, from flasks to bottles, was pressed into service to grow more mold, the source of their precious penicillin. The process was slow and inefficient, a far cry from the streamlined production methods of modern pharmaceutical manufacturing. It took gallons of broth, painstakingly cultivated and processed, to yield even a single dose of penicillin. Still, the profound promise of penicillin, its potential to save countless lives, drove them forward, fueling their determination. They had witnessed its remarkable power firsthand in mice, seeing the miraculous recovery from a deadly infection. And now they were resolutely determined to extend that life-saving potential to people, to alleviate suffering and offer hope where there was none. The race was on, a desperate sprint against time, to produce enough penicillin to conduct a human trial, the ultimate test of its efficacy. The world's first antibiotic was almost ready, poised to face its greatest challenge yet, the treatment of a human patient. The Oxford team's groundbreaking breakthrough had irrevocably set the stage for a medical revolution, a paradigm shift in the treatment of infectious diseases. The next patient, the one who would receive this experimental treatment, would be human, marking a pivotal moment in medical history. The stakes had never been higher, the weight of expectation pressing down on the shoulders of the researchers as they prepared to embark on this unprecedented trial. After the promising results in mice, the Oxford team was eager to test penicillin on a human patient. The weight of their hopes and the future of this potential wonder drug rested on this next crucial step. That patient was Albert Alexander, a police constable who was desperately ill, teetering on the brink of death from a severe, overwhelming infection. His condition was critical, and conventional treatments had failed. In February 1941, with time running out, he received the very first injection of penicillin ever administered to a human being. It was a momentous occasion, filled with both hope and trepidation. The results were astonishing. His dangerously high fever began to drop, signaling that the drug was indeed fighting the infection. The infected wounds that plagued him started to heal, a visible testament to penicillin's power, and against all odds, he began to recover. It seemed like a miracle. But the team quickly ran out of their precious, painstakingly produced supply of penicillin. Desperate to continue the treatment, they even tried to recover the drug from his urine, attempting to recycle every last precious drop. Despite their heroic efforts and ingenuity, the supply was completely exhausted, and tragically, Alexander's infection returned with renewed force. He died a few weeks later, a devastating blow to the team, and a stark reminder of the limitations they faced. The trial, which had begun with such promise, ended in tragedy, but it unequivocally proved penicillin's remarkable power to cure even the most resistant infections. The only remaining obstacle, and it was a significant one, was finding a way of making enough of it to meet the overwhelming need. The small Oxford lab, with its limited resources, simply couldn't keep up with the potential demand. The team realized that they desperately needed help, a partner capable of producing penicillin on a truly massive industrial scale. Britain, under constant siege and grappling with the immense strain of World War II, simply lacked the necessary resources and industrial capacity. And so, the urgent search for a production partner, one that could rise to the challenge, began in earnest. The future of this miracle drug, and the countless lives it could potentially save, depended entirely on finding a way to make more, much more. The next chapter in the penicillin story would take this life-saving drug across the vast expanse of the Atlantic Ocean to America. Albert Alexander's case showed penicillin's promise and its supply problem. Britain's war effort left little room for drug production. Florey and Heatley turned to the United States for help. In 1941, they traveled to America with a sample of the mold and their research. Their mission: convince U.S. scientists and industry to mass-produce penicillin. The timing was perfect. America was preparing for war and saw the drug's potential. At a lab in Peoria, Illinois, researchers improved the process, using corn-steep liquor to boost yields. Moldy Mary Hunt found a new, highly productive strain on a cantaloupe. Industrial-scale penicillin production had begun. After Pearl Harbor, penicillin became a top U.S. priority. Major pharmaceutical companies joined forces to solve the production challenge. The breakthrough came with deep tank fermentation, allowing penicillin to be made in huge vats. By 1943, production soared, and the drug was reserved for the military. On the battlefield, penicillin saved countless lives by preventing deadly infections. By D-Day, there was enough to treat every wounded Allied soldier. Mortality rates from infection plummeted. Penicillin gave the Allies a crucial advantage. The quiet work of scientists had become a decisive weapon of war. The age of antibiotics had truly begun. After World War II, penicillin was released to the public, sparking a medical revolution. Diseases that once killed millions became treatable. Life expectancy soared and child mortality dropped. Surgery became safer, and new procedures were possible. The discovery inspired a golden age of antibiotic research. Drugs like streptomycin and tetracycline soon followed. The pharmaceutical